Okay, it looks like we're live. Hi, everyone. Thank you so much for joining me today. My name is Dr. Danielle Snovlak, and I am the Senior Director of Education at Edvotec. And I'm here to welcome you to another one of our biotechnology training live streams. Thanks for spending the afternoon with me. Well, this hour, not the whole afternoon, but you know what I mean. For those of you who are new to Edvotec, we are the biotechnology education company. We were founded over 30 years ago by Dr. Jack Trixian, who was a professor of biochemistry at Georgetown University. And we were really born to translate innovation in the biotechnology labs around the world into experiences that your students could have in middle school, high school, and college classrooms. You know, there were so many fantastic things being done in biotechnology, but they weren't being used with students. And so, thus, Edvotec was born. And so we work with educators all over the world to demystify science and to foster the next generation of researchers through hands-on active learning activities. But we want to make biotechnology really accessible to everyone. And, that includes research labs, outreach centers, biohacker spaces, and of course, our teachers who we've worked with for many years. And so today we're doing a really fun experiment. It's one that needs minimal equipment in your lab. Really everything that you need to do this is included in the box. We are running an ELISA kit. We're running kit 279, which is investigating human health using the ELISA. So this is a test that's commonly used in medical and diagnostic labs around the world every day. The ELISA is the Enzyme-Linked Immunosorbent Assay. And it's a highly sensitive test that uses antibodies to detect the presence of specific molecules in samples. The test we're running today is a real, authentic ELISA in that it is using real antibodies to detect specific molecules within samples. But we're not actually using any samples that are infectious or biohazardous or potentially going to cause a problem with your students. We want all the reagents to be classroom safe, non-hazardous, but also we want your students to be able to perform an authentic ELISA. And the nice thing about using general reagents is that with this experiment, we include 3 different scenarios that you could use in the classroom with your students to be able to tailor the lessons to your curriculum. And you could even use this kit for something else that might fit better with your particular experiments. It's incredibly versatile in terms of designing scenarios. The lessons we're providing focus on common tests in medicine and in food production. And this would allow you to choose a scenario that works best with your curriculum. I'm going to walk you through the assay from start to finish over the course of this workshop. And while the different steps are running and our plates are incubating, we'll talk about how ELISAs work and the science behind each of the 3 scenarios that we present in this kit. This demonstration is being recorded, and our slides will be available on our website afterwards if you would like them. If you want to be notified when they're posted, please fill out the form. Maria is going to post it in the chat box. So it is a form that you please fill out, and we will then send you the slides, and we can send you a professional development certificate. But that link will only be live for an hour following this workshop. So please be sure to fill it out by 4 p.m. Eastern Time in order to get that professional development certificate. And the slides will always be available on our website with this experiment. So let's get started. Let's talk more about the ELISA. Maria, when you get a chance, can you post that link? Thank you. So as I mentioned before, the ELISA is a technique that is going to use antibodies to detect the presence of antigens in samples. Since we can generate antibodies to a lot of different molecules, the ELISA becomes a really versatile laboratory test that can be adapted for many uses. For example, if you have an allergy, you may need to be careful about what you eat. Foods that contain common allergens can be tested by ELISA to ensure that they are safe to eat. And we've listed a bunch of different applications for ELISAs in this slide here. Again, We'll talk more about pregnancy tests today. We'll talk more about the detection of proteins in foods. And, but a common place where you may be talking a lot about ELISA assays is in testing for COVID-19, which is the SARS-CoV-2 virus. And so the rapid tests that you may take at home or in a clinician's office, a lot of the rapid tests, if they are antigen tests, they are ELISAs. So we're using antibodies to detect the antigens that are on the surface of that virus. For this workshop, we're going to be talking about the ELISA that's used in medical testing and in food testing. The ELISA is commonly used for medical diagnostics, as it can be used to identify antigens in most body fluids and in other biological samples. And for example, if you have ever taken a pregnancy test, you have performed, or a COVID test, you have performed an ELISA in the comfort of your own home already. That's how simple, fast, and robust these tests are. Now, there are two main kinds of ELISAs. I like to think of them as quantitative or qualitative. And the overarching ways to think about these ELISAs is that a qualitative is a yes or no answer, and a quantitative is how much, right? And so a qualitative ELISA gives us a yes or no answer, and we can tell what the answer is based on comparing to a known standard. And so the one that I want a quantitative ELISA that you may be familiar with is the pregnancy test, which we'll discuss more later. This at-home test is going to test a person's urine for hormone that is an indicator of pregnancy. And if a person is pregnant, they will get a yes result. If they are not pregnant, they will get a no result. In general, these assays are pretty robust if taken within the appropriate time period. And it is a yes or no answer, because you can't just be a little bit pregnant. You either are pregnant or you aren't pregnant. And so that gives us a definitive answer, which is then confirmed with a more sensitive test in the doctor's office. Now, the quantitative ELISA is more sensitive, and that is telling us how much. And it can tell us how much by comparison to standards from result that we create from samples of known concentration. And so, if we have an antigen, a pure antigen in our lab, we can create a dilution series, and then we can use our antibodies to quantitate the intensity of the signal in each of those different conditions. And then we can use those to compare to our experimental sample to determine a concentration. And so one place where you might use this is with food allergens. We'll talk about gluten allergies later, but here is an example of a food label that you may see in the supermarket. And this is for a gluten-free product. It has been tested for the absence or presence of gluten using the ELISA. And one thing, this is a gluten-free oat. Oats do not contain gluten, but they're often manufactured in places where gluten-containing grains are present. And so what you do is you quantitate how much gluten is present in a sample. And if you look here on the label, what you can see is that the label does say in that little box, it does say it's ELISA tested to 3 parts per million. And so what you would do is perform the ELISA on this sample and create a standard curve. And where the sample meets the standard curve, that would be your concentration. And so this can tell us how much. And so the ELISAs we're going to be performing today are qualitative ELISAs. They are yes or no answers. But if you are interested in qualitative ELISAs, we do offer quite a few that you can perform. And so what do you need to perform an ELISA experiment? So that is our next big question. And so let's actually get started with the experiment. We're going to look at the reagents. And we're going to start doing the science. And you're going to see how fast and easy this is. So to perform an ELISA, everything you need is going to be included with this 279 kit. And Maria, if you want to link to the kit in the chat box, just in case anyone is interested, then they can go directly to the experiment. And so I'm wearing gloves. It's always good practice to wear the correct personal protective equipment. But again, there are no live samples, no viruses or infectious particles involved with our assay today. it is a simulation. And it's a real ELISA using real reagents, but it is a simulation of medical tests. So what we're going to do is I'm going to show you all the reagents and what they do. And the first one are going to be our antigens. And so we're going to include this is going to help us create the samples that we want to test. And so the samples we're going to test may or may not contain the molecules that we are looking for or the antigens. And using the ELISA, we can test for most biomolecules, pretty much anything the immune system can recognize. So we are actually, the antigen that we're using here, if you refer to the literature, we're actually using whey, which is a milk protein. And then we are going to use an anti-whey antibody, which is here. I've already opened these vials, but our antibodies and antigens come in our lipoprotein format, and you just add the dilution buffer to be able to reconstitute them, and it's really easy. You're going to take for example, you would take your antigen solution, you're going to add buffer, and then you're just going to move it to a 15 ml conical tube before aliquoting this for your students. Next, we're going to have two samples. We're going to have our experimental samples, and we're going to have our control samples. Our controls for our medical tests, our controls are going to allow us to know whether our experiment is working right. A positive control will give a positive result, and a negative control a negative result. If our controls fail, or if our assay does not give the correct results, it does represent a learning moment. In that we can have our students analyze and explain what went wrong as carefully as what went right. And so I think at this point it is important to acknowledge that science can be messy. These types of experiments do have opportunities for cross-contamination between the wells and the plate. But even experiments that don't work according to the manual are a learning moment where we can learn important lessons about data interpretation, of reproducibility, and other areas that it's important to emphasize with your students happen in science. Our experimental samples differ based on the test. For many medical tests, it could be blood, urine, or saliva from the patient. For food tests, this could either be raw materials or the final product itself. We don't know how they're going to turn out, whether they'll be positive or negative. So maybe you do if you're paying attention a few slides ago. I did have the picture. But we're going to go through pretending that we do not know which sample is which. We talked about the primary antibody. This is going to be the antibody that is going to detect. Oh, do I not have primary antibody on here? I do not have primary antibody on my slide. Oh my goodness. Primary antibody is going to be, I will amend that. If you decide to get the slides from me, I will amend that. The primary antibody, this is the antibody that is going to recognize our antigen. So like I said, if we have whey as our antigen, we're going to have an anti-whey antibody as our primary antibody. Our secondary antibody is our enzyme-linked antibody. This antibody allows us to detect the presence of our protein complex in our samples. And it's connected. The thing about this antibody is it's just a regular antibody. And it's an anti-antibody, meaning it is an antibody that recognizes antibodies, but it is conjugated to an enzyme. And this is important because that enzyme is going to allow us to turn over a substrate, in this case ABTS, and that is going to allow us to visualize our results. So your substrate will generally produce color or light. ABTS is going to produce color, and you will see that over the course of this experiment. You're going to need buffers. We have wash buffer and dilution buffer. These come with the kits. You do have to dilute your wash buffer when you get it into your classroom, but the dilution buffer comes at the correct concentration, ready to use. You have microtiter plates. You will have these pipettes. We're going to use these pipette. The microtiter pipette plate is like our little test tubes. Each of these is going to be a separate reaction. Our pipettes are going to let us transfer liquids. If you have adjustable volume pipettes, and you can use them for this assay just as easily. I'm going to use the transfer pipettes simply because not everybody has an adjustable volume micropipette. So I'll use what everybody will get with the kit. And then finally, and then that's what we need. So let's get started. we are going to work through this assay, this protocol together, step by step. Due to a little movie magic, I already have all of my samples aliquoted, and I already have all of my pipettes labeled so that we don't have to go through that step together. But you can see I have my plate here. I'm going to bring in my Post-its so that you can see them better. This is where we're going to talk. This is so you can kind of always know what step we're on. And then here are my pipettes. I have extra pipettes here just in case I accidentally contaminate something or if I drop one or lose it. One place where you could commonly get cross-contamination is using the pipettes. So we're going to do the first 3 steps here. The first 4 steps here, highlighted in red. The first thing we would do is prepare all these reagents. I already did that. And then using the appropriate transfer pipettes, I'm going to add 3 drops of each sample into the wells. So here are all of my pre-aliquoted reagents. So our first up, we have our positive control. Again, this positive control is going to contain our antigen. That is the molecule that we are testing for. And we're just going to put 3 drops in. 1, 2, 3. 1, 2, 3. 1, 2, 3. Let me get another drop. And you're going to want to make sure to have these pipettes labeled because we wouldn't want to take this positive pipette and put it into our negative sample because that will give us the wrong result. We will keep this sample to, remove the liquid now from our positive wells. And each one of these wells is almost like an individual, okay, that's my positive. Where's my minus? Here it is. There they are. Each of these little wells is almost like its own little test tube. So we are actually going to be performing 12 separate ELISAs over the course of this experiment. And so this is our negative control sample. This sample should not contain any antigens if I did my homework correctly. And if I don't cross-contaminate anything, which is, of course, always a possibility. Experimental error is a big part of, life in science. And sometimes we have to try things multiple times in order to get consistent, reproducible results without contamination. Here is our experimental sample 1. I'm going to drip those into here. 1, 2, 3. 1, 2, 3. 1, 2, 3. I'm sure there's nothing more exciting than watching me drip different proteins onto these wells. But the thing is with this experiment, you get the results so quickly and they're so robust. It's a really fun experiment and a really fun application of real science for your students to perform. So our 2 experimental samples, I know which one is going to test positive. You all do not. And so we will see by the end of this workshop where our antigens are in this plate. So right now, we're going to give this a few minutes to incubate. I'm just going to draw on a little bit of card real quick. What we've basically done is here's our little individual well, and we are adhering our antigens in here. So these are our antigens, and let's talk about what is actually happening in this well right now. First we're going to talk about our reagents. And so let's talk about antibodies, which are also known as immunoglobulins. These are specialized proteins made by our immune system to differentiate between self and non-self proteins or polysaccharides. And if anybody has any questions, please stop me. I am happy to answer the questions. You can either ask them now or at the end of the workshop. Make sure to give us a like or comment if there's any other questions or concerns. We are always happy to address them. So we're talking about these antibodies, which are these molecules that can recognize, that allow us to differentiate between self and non-self proteins or polysaccharides. We call these non-self proteins and polysaccharides antibody generators or antigens. Each antibody is a Y-shaped molecule composed of 4 polypeptide chains, 2 heavy chains, and 2 light chains. The polypeptides are linked together by disulfide bonds. Now, if we were to compare the amino acid sequence of 2 antibodies, the vast majority of the sequence is the same. The light chains are going to be similar. The heavy chains are going to be the similar. Let's say between an antibody that recognizes whey or an antibody that recognizes, gluten, or one that recognizes of albumin. And so all these three proteins, the light chains and the heavy chains are going to be mostly the same. However, if we were to compare the amino acid sequence at the antigen binding site, which is that little pocket at the end of the Y, we're going to see a great variability in that sequence. And this variability, the difference in its amino acid sequence, allows each antibody to recognize a unique epitope or a particular location within an antigen. Since the sequence can be so variable, antibodies can recognize a lot of different molecules. And so you can imagine the antibody-antigen connection like puzzle pieces. There are a lot of different puzzle pieces that are similar, but they only lock together when there's an exact match. And this marks the antigen. In the body, in the human body, the antibodies mark the antigen, whether it is an invading bacteria or virus or a cancer cell, for destruction by the immune system. Because of their specificity, antibodies can be used to detect the presence of specific biomolecules like proteins, peptides, antigens, or hormones in a complex sample. Oh, there's my puzzle pieces. And so all of those fit when your antibody and your antigen are going to fit together like a puzzle piece. So antibodies are produced when animals like rabbits, mice, or guinea pigs are injected with an antigen. Since many different immune cells within the body, and these are all animals with an immune system are going to produce this. When I talk about injecting a rabbit or a mouse, this is how antibodies are generated in the lab in many times. Antibodies are going to produce when when an antigen is introduced to the animals. Since many different immune cells within the animal produce antibodies in response to the antigen, the serum will contain a mixture of antibodies that vary in their ability to bind to our antigen. This mixture of antibodies is called polyclonal. If we isolate and culture individual immune systems cells from these animals, we can then create a monoclonal antibody, which is an antibody that recognizes only a single epitope within a protein. So we can think about our antigen like a water bottle. The hand in this picture is holding the bottom corner of a water bottle. We can think of that hand as an antibody that can recognize one specific antigen. Now, and that would be a monoclonal antibody. So we're only recognizing one surface of that bottle. However, if you took your other hand and held the cap of that water bottle simultaneously, we would be recognizing two sites on the water bottle, not just one. And so we could think of that as being like a polyclonal antibody in that it recognizes two different locations of our antigen. Now, one type of antibody isn't necessarily better or worse than another. Both types can be used in a wide array of experiments, from the ELISA to staining cells or doing a Western blot. The most important thing is that when developing an assay, to take the time and the care to select the best antibody for your application. To be used in the laboratory, antibodies must have specific, robust, and reproducible interaction with their antigen. Antibodies that have a high affinity for nonspecific antigens will have unwanted cross-reactions that can result in high background. In contrast, an antibody with a weak affinity may not be sensitive enough for antigen detection. And so these antibodies would produce results with a high positive or a high false positive or high false negative rate. All right. And so we're going to head to the next step of our ELISA. Our antigens have had time to stick to those wells, and we'll talk about that more in our next section. We're going to talk about what's happening in the well a little bit more in our next section. But what I'm going to do is remove all these samples. I'm going to put the waste in this waste beaker. And we want to be really careful to use the correct pipette for the correct well. So we're going to start here with the positive. And we're just going to remove all of this sample to our waste beaker. So that was our positive. Where is my negative pipette? Did I lose it again? Oh, I knocked it onto the floor. So there's something to look for. We're going to see how this works out, if there's the potential for cross-contamination because I knocked this on the floor. It's always good to recognize where you might have a little bit of error. Again, if we see error there, that is a place where you can have your students interpret what they did, what potentially went wrong, and how they would prevent it again the next time. I could prevent, knocking my pipettes over a million times, by, having a beaker to place them in, or by keeping them away from the edge of my surface. If I happen to use the wrong pipette for a sample, we could then say, well, to eliminate cross-contamination, there are specific steps that I would take. And all of these are important. It's important to know that mistakes happen, and how we can address them and how we can make our assays more robust in the future. So now what I'm going to do is I am going to wash with buffer twice. So I have our wash buffer, and I am going to place this wash buffer in all of these wells. And we're going to then remove it, and then we're going to put more in. So now is a great time to ask questions, because we are going to be doing a little bit of just making a mess and pipetting for the next few minutes. No, trying not to make a mess. So this is my wash. And what we're doing when we're washing, and we'll talk about this more in the next slide, but basically we are, almost grabbed the wrong pipette. We're going to take our positive pipette and we're going to remove this wash buffer. And the one thing, and we'll talk about this more in the next slide, is basically we are using the principles of attraction to stick protein molecules to the surface of these plates, right? So proteins have positive and negative regions. They're pretty sticky. Plastic is pretty sticky too. And so we count on the stickiness of the protein and the stickiness of the plastic to capture these antibodies on the surface of the plastic. We'll talk a little bit more specifically about lateral flow tests in a later slide. The lateral flow test is what you might be familiar with, with a pregnancy test or a rapid flu test or a COVID test, where basically you do spot a small amount of a sample onto, a strip, and then you see the lines turn color. Now, there are a lot of different kinds of ELISAs. We're doing a direct ELISA today. If you have interest in different kinds of ELISAs, we do have a workshop where we talked about the sandwich ELISA, which is a slightly different way of arranging your antibodies and antigens, but we're not going to talk about that in any depth today, just because we're just doing a regular direct ELISA. And remember, if you have any questions, please put them in the chat box. I love questions. But also, if you want the professional development certificate and the slides sent to you, be sure to put that in the chat, in the form as well, because that means I'll have your contact information and I can reach out to you. And there's a place on that form also where you can tell me if there's a specific workshop you want to see in the future, and I'll be happy to try and program it. We are going to be running some immunoassays if you are going to the National Science Teachers Science Teaching Convention in April. So you can even get your hands-on experience with some of these assays there. Okay, so I have washed my wells twice. Now we're going to add 3 drops of the primary antibody to each of the wells. And this is our antibody that is recognizing our antigen. Where did I put that? That's over here. So that antibody is going to recognize our antigen and stick to it. So here we go. And this antibody, we're going to add it to all our wells. It's going to be the same antibody. We're going to add like 3 drops in each. 3. You want to make sure to get 3 so that we have enough antibody to stick to all of our antigens. Here we go. I know you guys love to watch me move clear liquids between clear tubes. Need to get some colored liquids from CSI. So we have a question. Are antibodies sometimes produced by genetically engineered microbes or lab animals always required? And so that's a great question. And I'm not entirely sure. I do know that, many times we can take the antibodies and take the immune cells from animals, and then those are grown in culture, so they're not necessarily grown in animals, and to be able to culture them in animal cells at large quantity. I do believe that some of the reason why it needs to be produced in animals is because we don't know the way the proteins are going to have to, the sequence of the protein that would be necessary to bind to certain molecules. But I imagine that, in the future, there may be a way to create variable sequences and then look for what could bind in the test tube. And so that's a great question that I'll have to look up and learn more about myself. And then I can let you know as well. I imagine once the antibody is created in an animal, there are ways to culture it that are non-invasive, but the only way I know to make those antibodies the first time is by using the immune system of an animal. That's a great question. Thank you for asking. Okay, so actually now I have this. We are at the add primary antibody stage, right? So I'm going to draw my little schematic again, right? So here's our little test tube. We have our antibody, or antigen, I'm going to call it AG. And then we added our primary antibody. And so this is where we are right now. There are many protocols for performing an ELISA, but they're all going to rely on using these antibodies to detect the presence of antigen in experimental samples. And they're all going to follow the same basic principles. First, we add our sample, where it sticks to the plastic walls of our microtiter plate through hydrophobic and electrostatic interactions. And that's what I was saying before. Plastic is sticky, proteins are sticky. They stick together in the plate, and we don't actually have to. There are going to be ELISAs where you may have to pre-treat the plate or have some special coating on it. But in general, our ELISAs are simple direct ELISAs where you don't need to do anything special to the plastic plate once you receive it. After washing away any excess sample, in most ELISAs, the wells are going to be blocked with a protein-containing buffer that prevents non-specific interactions. And this is important, because antibodies are also proteins. And like I said, proteins are sticky and they can stick to the wells. And so if we didn't block the wells, we would get antibodies sticking to the plastic of the wells, and that would give us false positives, right? Because we would have the presence of the antibody there without actually having our antigen present. And so we would do something called blocking. But for the ELISA that we're performing today, we've simplified it so that we don't need the blocking step. So there are blocking reagents in our other samples so that you can block and bind at the same time, and that does save time. We then add the primary antibody to each well. If the antigen is present, so you can see it here and you can see it you can see it in my little drawing and you can see it on the screen, where the antigen is represented by the green molecule that is stuck to the little well, and our primary antibody is the pink molecule. In cases where we have the antigen, that pink molecule fits together like a puzzle piece, and they adhere to one another through electrostatic interactions. We would then wash any excess antibody out of the wells. And then in our ELISA, our antibodies, are going, as I mentioned before, we're using a whey protein as our antigen, and we're using an anti-whey antibody. So in this ELISA, our antibodies come from an animal that has been introduced to the whey protein, created antibodies to whey, and now that interaction is happening in our tubes. The next step would be then after washing, we're going to have washing between most of these steps to, make sure that there's nothing extra hanging out. And then we're going to add our secondary antibody. Our secondary antibody recognizes our primary antibody in the solution. If that primary antibody antigen complex is formed in the wells, our secondary antibody will then bind on and form part of the complex and remain in that well after the samples are washed. And you may be thinking this is kind of crazy, like why are we using multiple antibodies that recognize each other? And this is actually a really great trick for these assays because there are two reasons that we do this. The first reason is an amplification effect. So we have our primary antibody. Here we're kind of showing the secondary antibody as being like monoclonal and that it's binding one place. But generally the secondary antibodies we're going to use are polyclonal. So they'll not only be able to recognize the top part of that antibody, but the sides of it, the other parts of it. So each primary antibody will have multiple secondary antibodies binding. So for every one antigen and one primary antibody, we have multiple secondary antibodies. We then conjugate this enzyme to our secondary antibody. And this is important because this is a chemically complicated reaction in getting those two proteins to bind together. And the nice thing about using a secondary antibody is that, again, it's going to recognize many locations on that primary antibody, but it's going to recognize any primary antibody from the same animal. So if we're using a mouse antibody in this case, the secondary would be an anti-mouse antibody. And anti-mouse antibodies could be raised against chicken, against egg, against milk, against any kind of antigens you could imagine. So we could have a whole library of primary antibodies that we could use in the lab, but we'd only need one secondary antibody that's linked to that enzyme to be able to detect all of those different things. And so it's great because we don't have to have as many specialized reagents to have a very powerful assay at hand. The second is an amplification effect. So the more antibodies we have bound, the better. And that will help us when we actually add our substrate to the wells. And so in general, we add a clear colorless substrate to each well. And in wells where we have that secondary antibody present, the enzyme is going to turn the clear substrate solution to blue. Since most enzymes have a high catalytic activity, meaning that they can quickly turn over the substrate, this assay allows us to quickly detect even the smallest amounts of antibody, right? Because we're having one antigen, one primary antibody. The primary antibody is recognized by multiple secondary antibodies. Each of those secondary antibodies has an enzyme connected to it, and it's constantly being able to turn over the substrate. And so you can imagine that by building that complex, we're actually, even if there's only one antigen in here, the potential, we have one antigen, maybe we have a dozen secondaries antibodies present, and then we're just constantly turning over that substrate because of the enzyme. And so that will allow us to be able to detect even vanishingly low amounts of our antigen in solution. And so if we think about like that gluten assay I was talking about, that's looking down to like 3 parts per million, which is vanishingly small. So let's think about I like to think about, when I think about the ELISA protocol, I like to think about the complex that we built like an ice cream cone. So the cone is like our antigen, which binds to the microtiter plate. Each scoop of ice cream, is an antibody in our complex. Our first scoop is our primary antibody, and the second is our enzyme-linked secondary antibody. And we're kind of building a complex on top of our antigen, much like we're building, a delicious dessert here by putting scoops of ice cream on top of the cone. All right, so we've had enough time for our primary to bind. Now we are going to wash the primary out of our wells. So again, the first thing we got to do is get the sample out of the wells. So I'm going to make sure to use my appropriately labeled pipettes here to remove all of this solution. And again, if you have any questions, now is the perfect time to ask, because I am going to be moving a lot of clear liquids from place to place. Again, trying to use the correct pipette to prevent any kind of cross-contamination. Again, I always like to make sure that, everyone knows that science doesn't always work the first time. That's the reason why people spend years in the lab trying to perfect techniques. But by the time you get some of these commercial ELISAs at home, they're going to work 95 to 99% of the time. And that is pretty remarkable, to be able to design an assay that works that reproducibly every time you do it at home. And that's by people who haven't trained to use it. I think about, I am a parent, I have 2 kids, and I think about, when you're taking a pregnancy test, you're really not thinking about performing it properly. And so it's just amazing that those work as well as they do, without any sort of technical expertise, in considering the way that some techniques can be finicky. I know we are some people watching with me. are you doing ELISAs in the classroom, and how are they working for you? We'd love to hear, how they're working for you. So right now I am removing my first wash. Again, that's going to take anything that's not bound to the complex in here and wash it out, because if we leave anything behind, that has the potential to start forming that complex, and that is something that we do not want to happen. So I'm removing the wash from my negative control, and then I'm going to have sample 1 and sample 2. I really like doing ELISAs with students. Again, they require minimal equipment. So everything you need to do for this ELISA is going to come in the box except for water. And even that is, it's like under 200 mils. So you could, like, if you don't have purified water in your classroom, you could literally just get a bottle of water and that water will work for you. All the plastic is included. Which is great because sometimes it's not included and then you have to bring on that extra and you don't realize it until you get it into your classroom. And that can be a problem, right? You're ready to do an experiment and you're missing something because it wasn't in the box. And that's really frustrating, when you have to pay extra for some of the components that are important. And so one of the things I always like about the experiments that we offer is if it's not in the box, we tell you right up front that you need it. And most of the time, the only thing you need, it's like just add water for most of our kits and equipment. Obviously, you need for some of our experiments that you're going to need the equipment. So we're going to remove this second wash. Again, this is removing any excess primary antibody. Yeah, this is removing any excess primary antibody from our wells. Again, if that primary antibody is sticking around where it's not supposed to be, that becomes problematic because the secondary antibody recognizes the primary antibody. It doesn't recognize the antigen. And because I'm constantly talking and I'm trying to talk and pipette at the same time, especially with these ELISAs, there's a pretty high probability that I mess up. We have a Nicole is saying that she hasn't done ELISAs, but someone gave them a kit. So what I would say is definitely give it a try. It is a perfect test. We do have a forensic, I believe we have a toxicology kit on our website. You could take a look at that literature to see whether it would apply to what you're doing. Offhand, I do not know specifically which drugs are analyzed by ELISA. Let me just explain what I'm doing next and then I'll come back to your questions. So this is going to be our secondary antibody. Remember, this is the antibody that's a polyclonal. It's going to recognize multiple sites on our primary antibody as we're setting up this complex in our mini test tubes. So I'm going to add this secondary antibody to all of my wells. If I can find the right pipette, this is my ABTS. Here's my secondary antibody. So what I would say is that I don't know did I use this already? I see some residue in my secondary antibody pipette. I'm just going to grab another one just so I don't make any mistakes. What I would say is if you're looking for a specific test, like a specific ELISA to use as a simulation, I would take a look to see what tests are specifically ordered by a county coroner in suspicious situations. I do know that there are ELISAs for a lot of the most common drugs, and then that these ELISAs are used to analyze biological samples. You will see some done using GC mass spec. And I think that happens when you need to have like a very quantitative amounts, and that might be what is done for an autopsy, where you want to figure out if something is a byproduct or if it's actually present, in intoxicating levels or in like murderous levels. The best place to start looking is maybe crime scene, like forensic science labs, or perhaps, a county coroner's office or FBI to kind of see like what they're testing for regularly. And even then, if a death is not suspicious, most of the times I don't think that anyone would look for anything out of the ordinary. It would be, if a person were, found in a compromising situation where you would then perform some of these tests. And Maria put up our forensic toxicology kit. I don't think that one is an ELISA, but it would give you a good background about toxicology from a forensic context. Yes, definitely can't afford a mass spec, GC mass spec, and most places can't. And I do think that the ELISA is a great way. Even this ELISA that we're doing, you could potentially use in a crime scene background, again, because the reagents are very general. It's when you apply the scenario that it becomes specific. All right, so I'm going to update our little graphic up here for adding our secondary antibody again. So here we have our little test tube. I'm going to continue to make it larger. We have our antigen. We have our primary antibody. And now we're adding our secondary antibody. It would actually kind of look more like this, right? So here is our antibody. And then we're going to have another antibody, our enzyme-linked antibody, hanging off of that. And maybe this is going to recognize all these different locations on this antibody. Okay, so while this is going, we do use the ELISA in 195. So if you look at the description, thank you, Maria. I never remember the specifics of every experiment, but you'll use the ELISA to analyze simulated crime scene samples for the presence of drugs. And so that'll be a great lip for you to review, to think about the ways that we can use ELISA, for drug detection. So here we're going to cover our 3 ELISA scenarios, and that is going to be, our first is going to be a pregnancy test. Again, this is an ELISA that many people may have taken themselves or been present when one was taken. And so this is a test, this is a test that helps you to determine early pregnancy. Some pregnancy tests work by determining the level of the human chorionic gonadotropin, or HCG hormone, in blood or urine. If you're doing it at home, it's likely going to be urine. HCG is a hormone that is produced by the placenta beginning very early in pregnancy, and it is essential for maintaining a successful pregnancy. Since HCG is only produced in the developing placenta, except in very rare cases where it might be secreted by a specific kind of cancer, that is one way that it's very indicative of a pregnancy. And since it's because it is detectable within a few days of implantation, it's a very specific and early indicator for pregnancy. HCG is a heterodimeric protein. That means it's made of 2 different subunits. Our alpha subunit, which has a molecular weight of 18 kilodaltons, and the beta chain, which has a molecular weight of 32 kilodaltons. Furthermore, this hormone is a glycoprotein, meaning that the protein has been covalently modified with specific protein and carbohydrate side chains. And so one thing that I actually found really interesting when I was doing the research for this workshop was that several glycoprotein hormones like HCG, but also like luteinizing hormone and follicle stimulating hormone, they all share the same alpha subunit. And so that means that in pregnancy tests, the antibodies used have been selected to detect only the unique beta subunit of this protein. So it's just shocking to me that something that you do at home can be that specific. That's why I love science. And so pregnancy tests detect levels of beta HCG in blood or urine samples. And so the pregnancy tests we use at home are lateral flow tests, and they detect our HCG hormone in urine. So when you pee on the stick, the biomolecules in your urine and in the water, it's going to travel through the test paper strip. And the beginning of that test paper strip is infused with our primary antibodies, right? So just like here, we put our primary antibody in first. In the lateral flow test, your biomolecules are going to meet up with that primary antibody first. If HCG is present, it's going to bind to the anti-HCG antibodies in this area. The flow of the sample then moves the molecules and the free and unbound, the molecules, the bound HCG and the free unbound antibodies through the test strip to zone 2. This area contains a second antibody, which is a detection antibody that also binds to HCG. And so what you can see here is when the complex is formed, the test strip changes color. Any free remaining antibody is going to continue traveling through the strip where it is going to bind to a controlled detection antibody. And so if this line doesn't change color, your test is invalid. You're going to want to take it again because that means that it's not working correctly. And this is the principle for most lateral flow tests that you might take, right? So you apply your sample to one end, as it travels through the strip, it then meets up with your primary antibody, your detection antibody, and your control antibody. And so one fun fact for your students, if you definitely request the slides if you want this link, but the first pregnancy test involved injecting frogs with urine from women and pregnant people to see whether they would lay eggs. If there was HCG present in the urine, it would trigger a hormonal reaction in the frog, and the frog would lay their eggs. And it was actually pretty accurate. So if you want to learn more, make sure to get these slides and then check the article on the pregnancy test from The Atlantic. Our next scenario, and I'm going to talk really quickly because we're getting close to the end. Our second scenario focuses on heart attacks or myocardial infarctions. They're one of the leading causes of death in the developed world. The most common symptoms of a heart attack include sudden chest pain, shortness of breath, sweating, fatigue, although many patients only experience minor symptoms. Most heart attacks occur due to a blockage of fat and cholesterol within a coronary artery, leading to reduced blood flow and oxygen to tissues over time. Eventually, the amount of blood traveling through the arteries is reduced to the point where it can no longer support the heart cells, and a patient will have a heart attack. The oxygen-deprived heart cells die, and unlike cells in many other tissues, they don't really regenerate themselves. They can't grow back. And so this results in a permanently scarred area on the heart, which can lead to additional complications in the future. Injured heart cells undergo the process of cell death, where they rupture and release their cellular components into the surrounding area. This can injure neighboring cells, which then increases the area of damaged heart tissue. In addition, the dying heart cells will also release proteins that can enter the bloodstream and travel throughout the body systems. But while this can be, bad for a person's physiology, it is useful as a medical test because doctors can perform blood tests to monitor levels of cardiac proteins like troponin and creatine kinase for evidence that a heart attack has taken place. So rapid detection and treatment of heart attack patients is critical to manage heart disease, minimize damage to the heart, and to prevent further complications. And so the test we're simulating is for troponin, which is a protein complex that is important for muscle contraction in the heart muscle and in the skeletal muscle. And there are specific types of this protein that we can look at that are indicators of the damaged heart muscle. So a person suffering from a heart attack will have a damaged heart muscle, which is going to lead to elevated amounts of this protein in the blood. This increase is going to start within 2 to 3 hours of the heart attack, peak in approximately 24 hours, and then persist for 24 weeks. And so in a clinical setting, we would test for this protein to determine whether or not a person is having cardiac symptoms. And it's important to note that these markers are a marker of all heart damage, not just a heart attack, which is obviously the most severe form of a heart disorder. And so an elevated troponin level can also identify patients who might be at risk for cardiac events as well. And elevated levels have been associated with an increase in cardiac mortality rate. Okay, so the third one is we're going to talk about food safety. I'm going to go through the real fast. Here we're talking about gluten. It's a structural protein found in grains like wheat, rye, and barley, and it can comprise about 80% of the total proteins found in these grains. And typically, it is going to be produced by enzyme. It's going to be processed by enzymes in your GI system. It's a major structural protein, and there are two main proteins in gluten, gliadin and glutonin, that together form a network in our baked goods like bread to make it light and fluffy. And if you want to learn more about gluten, I've linked to a really great article. It's actually written for a high school audience, so it'd be perfect for your students to learn more about gluten protein and actually about its effects on the body. But not every person can tolerate gluten in their food. And so this is where disorders like celiac disease, play a role. And so celiac is a serious immune disease that affects the gastrointestinal system. and it affects approximately every 1 in 141 Americans. And in patients with celiac, dietary gluten triggers an immune response that can lead to pain, bloating, and severe damage to the small intestine. And so this is actually really difficult to diagnose because many of these symptoms are shared with lactose intolerance, IBS, or Crohn's disease. And so there are processes that doctors will take to be able to diagnose this. But patients with celiac disease must carefully regulate their diet to avoid gluten. And this means the patient must completely remove all gluten from their foods and drinks they consume. So there are even gluten-free beers because gluten in beer can cause a problem. There's gluten-free breads and gluten-free foods. Even cosmetics can contain gluten. And so a person with celiac has to be really careful because some can be triggered by even the smallest amount of gluten in their products. And so due to the number of people who are requiring gluten-free diets, many food products and many consumer products are now tested to ensure that they are gluten-free. The US FDA is going to require that foods contain less than 20 parts per million gluten, but while many celiac support organizations require even lower levels. And so if you are a person with a gluten intolerance, this is something definitely to discuss with your doctor as to what is an acceptable level. All right, so we are going to power through these last steps really quickly. I am going to remove our secondary antibody. I am going to wash really quickly, and then we are going to visualize our results. So let me remove this secondary antibody. And this is a step that's really important to wash really well because we do not want any of that extra secondary antibody around. Because if we just had secondary antibody and added our antigen, what do you think is going to happen? Basically, any antigen, any antibody, secondary antibody is around, is going to be able to react with our substrate. Is going to be able to react with our substrate. So now we're going to see how good I am at keeping all these samples separate. But trust me, like once we get the substrate in here, our color change reaction is going to happen fast because it's actually very warm in my office. And the heat actually is going to increase the rate of our enzymatic reaction. So this is my first wash. Again, we're going to be careful not to cross-contaminate between samples. If you overfill the wells, they could potentially, run into one another, which would be an area where we could potentially have cross-contamination. Again, cross-contamination is sometimes a part of science, as much as we don't like to admit it, and that's why we need to use good lab practices to reduce the amount of cross-contamination. That's why we have control samples. If our control samples show signs of contamination, that shows we did something wrong and the results of our assay may not be able to be trusted. I have my S2 pipette, so I'm going to go to S2 instead of S1 this time, just because I already had it in my hands. The ELISA is very simple, very powerful, probably if I wasn't talking so much, we would have been able to do it even faster than I did it. Usually I feel like you can do this assay in about 40 minutes if everything is set up right. The biggest time is the incubation times, in that, we got to wait for all of our complexes to form. But even that said, to be able to finish an ELISA in a class period, to be able to do an experiment like this in a class period, I think it's a really powerful assay, a really powerful thing for students to be able to do. And I know I am always a fan of hands-on experiential learning whenever possible. And if this is a pretty straightforward example of an ELISA, we do have a more inquiry-based ELISA. The one that I really like is we have a quantitative ELISA where students can actually, it looks for whey. It's going to use these same antibodies, and it's going to look for milk proteins. Milk protein is a really common allergy, especially in young children. And so you have to make sure that food products don't have milk in it in order to prevent that allergic reaction. And so the test actually allows students to bring in different food products and test them for the presence of whey. So we're going to add our substrate now. This is going to be turned over by our secondary antibody. And in samples where it's positive, we should start to see a bright green color. enter in the wells. It's going to take a couple of minutes to generate. I am putting too much substrate in here. Let me grab more substrate. I'm moving quickly, and I put a little too much in each of my wells, which is no big deal because I have a lot of it left. And I will just use this here. So if you're using these reagents, the primary antibody and the antigens can be reconstituted pretty early. Your secondary antibody and your ABTS solutions, you're really going to want to use them within 24 hours of prep. Like we've had success with them for as long as 5 days. But basically what you would want to do is test them first before you do it with your students. You'd add your secondary, a small amount of secondary to a small amount of ABTS and make sure you get that good color change. So if you look now, let me bring the camera down even more. You should be able to already see our green color change. And as you're watching with me, you'll see it's going to get darker and darker. I mean, look at the difference between our positive control and, the difference in the intensity in the green color. Okay, so we've added our substrate. This is basically a schematic of our results. You can see I mislabeled something a little in that I put our negative control second instead of our negative control first. But what you can see is that I did not cross-contaminate my samples, which is amazing. Our negative control should be clear. After performing this assay, that means there's no antigen present, where our positive control, again, is going to be green. It is going to be positive. The sample that I've set up is sample 1 will be clear, no antigen. The sample I've set up for sample 2 is going to be green, which means there's the antigen present. But we give you everything and you can customize it for your class. Maybe you want to have every student have different results. You could give them a different combination and then the students could analyze the data set as a whole. I hope you all can see, it looks pretty good on my screen, but you should be able to see a very clear color difference between the positive and the negative samples. And I'm very proud of myself for not cross-contaminating. Just going to put that out there. So if you have any final questions, please ask them now. I'm going to wrap up. Fill out the form that Maria posted earlier. I will send you the slides. They'll be annotated. And if you're here, I will send you a professional development certificate saying that you were here with me for this workshop. We've come to the end of this experiment. You've listened to me talk about a lot of different information while we've been together. We talked about the ELISA and how this really simple and effective technique can use antibodies to detect different antigens in solution. And we also explored some common applications for ELISA technology, including many that are important for our daily lives, like pregnancy tests, heart attack tests, and gluten tests. Because ELISAs are so common in the lab for so many different conditions and applications, and like Nicole, you mentioned, forensics uses a lot of ELISAs, forensic science, criminology uses ELISAs a lot for different agents. This is a perfect way to teach your students about these kinds of different testings that go on just in the context of life. Your students can think about how ELISAs are used in our daily lives now for rapid tests or COVID tests. And you could even have them think about questions in public health, questions in food safety, and how we could apply the ELISA in the future. Your students are likely curious to learn more, and they can do the research to design a new application for the ELISA. I will be posting these slides in the next few days to our website. If you want me to email you when they're available, please fill out Maria's Google form link. That will give me your information so that we can contact you. I will not spam you with anything else, so don't worry. Just be sure to do that. Be sure to like this video and comment on it if you found it useful. And on this slide are all the ways to get in touch with us at Edvotec. We're really responsive. We pride ourselves on customer service, and we really want to figure out ways to make sure that you are having a great biotechnology experience in your lab. We're on Facebook, we're on Twitter, we're on Instagram, and we're very recently on TikTok, which has been a lot of fun. Even though I am not clearly I am not of the TikTok generation. I have been enjoying it. So thank you so much for your time. We're so glad that you were here to spend this time with us, to learn about the ELISA, to talk about biotechnology. And we're really happy to be working with you to get biotechnology techniques into their classrooms, because I think they're a really fun way to get students engaged with scientists. with science. So thanks again. Have a fantastic day. Enjoy it's a Thursday, we're coming up on a weekend. So make sure to take some time to rest, relax, and recharge, and then do some biotech. Have a great day, and we'll talk to you soon. Bye now.